to study about animal kingdom, types of eggs. Based on the quantity of yolk present, the eggs of animals are broadly divided into three types, namely microlecithal, mesolecithal, and megalecithal. Microlecithal eggs. They contain a very small amount of yolk. Example: the eggs of sea urchins, Herdmania, Amphioxus, and most of the mammals. The eggs of human beings contain almost negligible amount of yolk, hence they are called alecithal eggs. Mesolecithal eggs. They contain moderate amount of yolk. Example: the eggs of lampreys, lungfishes, frogs, and toads. Macrolecithal or megalecithal or polylecithal eggs. They contain a large amount of yolk. Example: the eggs of insects, sharks, bony fishes, reptiles, birds, and egg-laying mammals. Based on the distribution of the yolk in the cytoplasm, the eggs of animals are classified into four types. They are homolecithal, telolecithal, myolecithal, and centrolecithal. Homolecithal eggs. The yolk is uniformly distributed in the cytoplasm of the egg. Example: the eggs of annelids, mollusks, echinoderms, and protochordates. Telolecithal eggs. The yolk is concentrated in the vegetal half of the egg. Example: the eggs of amphibians. Myolecithal eggs. The yolk is abundant and occupies nearly the entire cytoplasm, leaving only a small disc-like area of the cytoplasm for the nucleus. Example: the eggs of reptiles, birds, and egg-laying mammals. Centrolecithal eggs. The yolk is localized in the center of the egg. Example: the eggs of insects. Types of cleavage. Cleavage is the process of mitotic division of the egg leading to the formation of a multicellular embryo. Cleavage is triggered by the activation of a cyclin-dependent kinase complex. The cells derived from cleavage are called the blastomeres and form a compact mass called the morula. Cleavage ends with the formation of the blastula. The type of cleavage depends mostly on the amount of yolk present in the egg. Determinate cleavage. In this type of cleavage, the fate of the blastomeres is set or determined in the early embryonic stages. This type is found in the protostomes. Indeterminate cleavage. In this type of cleavage, the fate of the blastomeres is set or determined relatively late in the embryonic development. This is found in the deuterostomes. Holoblastic cleavage. In this type, the cleavage planes pass all through the egg. It occurs in the microlecithal and megalecithal eggs. Meroblastic cleavage. In the megalecithal eggs, due to the large amount of yolk, the cleavage planes cannot pass throughout the egg. Two types of meroblastic cleavages are observed, namely discoidal and superficial. Discoidal cleavage. It occurs in the megalecithal and telolecithal eggs. The cleavage planes do not penetrate the yolk. The embryo forms a disc of cells called the blastodisc on top of the yolk. It is found in the reptiles, birds, and the egg-laying mammals. Superficial cleavage. It occurs in the centrolecithal eggs. The cleavage involves mitosis, but not cytokinesis. The nuclei are formed at the periphery, and the membrane grows inwards, partitioning the nuclei into individual cells. It is found in the insects.